Hello, my name is Pim Fredericks, and I'm a PhD student at the University of Strathclyde, joined between the groups of biomolecular and chemical physics and pure and applied chemistry. Today I will tell you something about my project entitled Building with Proteins, so you can expect a hint of biology, chemistry, and of course physics. Let me start by giving you a brief introduction about this protein building. It has been shown that various short peptides, that means proteins of only a couple of amino acids, can self-assemble into various shapes and sizes of nanostructures. You can see some examples here, all of which are smaller than a micrometer. On the left you have a fibrous network that can extend into 3 dimensions. In the middle you can see that you can create all kinds of beautiful structures like flower-like assemblies. And on the right you can see a nanotube of only 100 nanometers in width. These materials have all kinds of applications. For example, you can use conducting proteins to make wires that can conduct electricity, which would be very useful in the case if you want to interface electronics and biomaterials. Because these materials are so bio-inspired, they will not be rejected by living organisms. So you can even use them for stem cell research. It's already been shown that if you use a material that's very stiff, you can actually control the stem cell into going into stiff tissue as well, like bone. Whereas if you use a soft material, then you can make them go into brain tissue or fatty tissue. A third application is responsive biomaterials, where you can use enzymes or even just small changes like the acidity of the environment to make this material change its properties. This can be very useful if you want, for example, to use it for drug delivery. On the other hand, if you break these structures, they can self-repair because they are held together by interactions that can heal themselves. The only thing that we don't know is how these materials actually work. So the goal of my project is to study and control the self-assembly of peptide materials. And I plan to achieve this with a threefold approach. First, we use computational methods to see if we can predict in a computer if a certain material will actually form these structures. Second, there's microscopy, which you can use to study the materials after they have been formed and get these images like in the previous slides. And third, the most physical approach is spectroscopy, where you use light, usually from lasers, to control or study the materials that are forming. I will show you some initial results of my project, and this is from the computational side of things. So in the experiment you can see diphenylalanine from the tubes that you saw 2 slides ago, and we've tried to simulate this using a computer. So you can see a couple of snapshots of the simulation here. On the left you have the starting point, where you can just see randomly oriented peptides in a cubic box. But if you then run the simulation, you can see that they actually come together, form a cluster, and the longer you go, you form vesicles first, and then in the end you have hollow tubes that are forming. So it's very beautiful that you can see that the experiment which shows tubes actually agrees perfectly with the computer model. So if you extend this to all kinds of molecules, you can hopefully use this method for screening all kinds of materials, because there's so many materials that you can make. It is impossible to test them all in the lab, and therefore it's very useful to use the computer to screen which molecules would be interesting to study. Second, also together with the microscopy and the spectroscopy, we hope to be able to get mechanistic info from this assembly process. So in many publications about this topic, you see these nice cartoons like on the right, where you have an enzyme somehow building a helical fiber by assembling lines and arrows. But this is of course not what really happens, and we don't know how it exactly works on the molecular level. I hope to be able to find out which interactions are important and how these molecules are actually coming together to form these structures. And if you understand something, the step to controlling something is usually not that big. And if we can control these assemblies, you can, of course, use them to your own advantage. In the end, just to sum up, very small biomolecules can self-assemble into various nanostructures with all kinds of applications. And if we study these molecules, we can hopefully understand and control them to use them to our own advantage. And thank you very much for listening.